Hi everyone, welcome back to our lectures. We are in the middle of chapter 12, talking about our endomembrane system and protein trafficking. We're about halfway through. We're talking about exocytosis and endocytosis. These are two ways that we move materials across the plasma membrane. And exocytosis, think of that word ex meaning like exiting. We are sending things out of the cell. Things are exiting from the cell. In endocytosis, we are bringing things into. So endo means in. We are bringing things from outside the cell into the cell. So here, something is moving from outside of the cell, in. That is endocytosis. Or if you want to do it backwards and move it out, that would be exocytosis. The ways that we move things from our cell to the exterior is called a secretory pathway because we are secreting things from the cell. And in our secretory pathways, proteins move from the endoplasmic reticulum through to the Golgi complex. They're moving through vesicles called secretory vesicles and granules called secretory granules. Then those granules fuse with the plasma membrane and send their contents to the exterior of the cell. So after these vesicles move from the trans-Golgi network, some of them can move right to the cell membrane and immediately release whatever they have in them. This process is just going to constantly be going on all the time. It doesn't need any sort of signals. This is called constitutive secretion. Constitutive means always on. You may have heard of constitutive genes or other things in your body that are constitutive. They are constantly doing it. So constitutive secretion is always active. One example would be within your intestinal cells. They are constantly secreting mucus into the intestinal lining. They are constantly secreting different enzymes for digestion. There's also some current evidence that there may be some tags that are required for enzymes to be incorporated into constitutive secretion. There's also regulated secretion. In regulated secretion, a vesicle leaves the Golgi complex and it's just kind of stored in these vesicles until a specific signal is released. And when that specific signal is released, here a chemical is binding to the cell that allows these molecules to be secreted. This is regulated secretion because we're going to regulate how it happens. It only happens when we send that signal. One example of this would be a neurotransmitter release. So you don't constantly want all of your nerve cells firing. Neurotransmitters need to be released under controlled sequences to get a trustworthy signal through your brain. And regulated secretory vesicles will form by budding from the trans-Golgi network as immature secretory vesicles, and they'll move closer to the site of secretion and just wait there. That signal could be something like an increase in calcium ion concentration that will trigger vesicles to release their contents. The vesicles will fuse with the membrane, and the contents will then be sent outside of the cell. In a lot of cases, a lot of cells will do what's called polarized secretion. So polar means the two sides are different. So they're only going to release their contents to one specific side of the cell. So if we come back to our intestinal cells here, here's the cells making the lining of your intestines. Along the apical side, the cells are facing your digestive tract. This is your intestines going through here. So these little villi are pulling in nutrients from your food. On the side, you've got, through the basolateral side, you've got connective tissue, and underneath that, your blood systems, blood vessels. If you are secreting digestive enzymes, you want that to come to the apical side into your intestines to break down the food. You do not want your digestive enzymes coming down and breaking down your cells and tissues on the basolateral side of the cell. That would cause a lot of damage. So this is polarized secretion. We carefully secrete our digestive enzymes only to the side of these cells where they're interacting with the intestines. In exocytosis, anything that's within a protein vesicle, or any proteins that are within a vesicle, can be released outside of the cell as that vesicle fuses. Some things that animals secrete would be hormones secreted by different cells, mucus, milk proteins, digestive enzymes. Plant and fungal enzymes can also be secreted through their exocytosis. They can also secrete structural proteins to make their cell wall. During exocytosis, first the vesicle is going to carry its products and move towards the surface of the cell. Then the membranes of that vesicle are going to fuse, connect here with the membranes of the cell membrane. Then as they fuse, the contents within the vesicle can be released, and the membrane becomes part of the plasma membrane. However, what was inside of the lumen of the vesicle now becomes the exterior side, the extracytoplasmic side of the cell, and whatever was on the outside of the vesicle now becomes the intracellular side of the membrane. So when the vesicle fuses with the plasma membrane, the luminal or inside membrane of the vesicle becomes part of the outer surface of the plasma membrane. And so any glycolipids and glycoproteins that were formed in the ER and the Golgi lumens will face the extracellular space. It's really important to have them facing the right way so that they can interact with other cells and be used in cell-to-cell contact and cell-to-cell interactions appropriately. Most eukaryotic cells are going to carry different forms of endocytosis, which allows you to bring things in. In endocytosis, a piece of the cell membrane is going to fold inward and then pinch off, which forms an endocytic vesicle. And within that endocytic vesicle are different substances that your cell just ingested. Most of these vesicles are going to develop into early endosomes. Those early endosomes can fuse with vesicles from the trans-Golgi network, where they pick up digestive enzymes and create lysosomes. In phagocytosis, you've picked up big solid particles of food. If you're something like an amoeba, phagocytosis is how you get food. And if you're multicellular, usually you only have certain cells that do this. Those certain cells that do phagocytosis are called phagocytes. Within our bodies, our phagocytic cells are pretty much restricted to cells of our immune system. Endocytosis can also involve pinocytosis, where just fluid is taken into the cell. Endocytosis is involved in a lot of important functions in our cell. One is maintaining normal cell homeostasis, bringing in products that we need, bringing in nutrients, recycling cell receptors, so any receptor that's here on the surface of your cell can be brought back in, protecting from bacterial invasion by phagocytosis of your white blood cells, communication with other cells. It's how we pick up if another cell is sending a signal. It gets picked up here. And also, some viruses have been able to co-opt this process to increase their infectivity. So a virus can bind to a receptor here, trigger your cell to say, I need to bring that in, cause endocytosis, and now you've got viruses in your cell. In humans, we have two kinds of white blood cells that are primarily the phagocytic cells that will protect us from bacterial infections. These are our neutrophils and our macrophages. And here, this bacteria has just invaded your body. So your neutrophils and your macrophages got that signal, there's an infection, go find it and kill it. That white blood cell will now come, find your bacteria infection, and it just kind of wraps its little plasma secretions around that cell. So it eventually forms this pseudopod, and that phagocytic vesicle comes in, fuses with lysosomal proteins, and breaks down that bacteria. In your human immune system, phagocytes create toxic amounts of oxidants that will kill microorganisms in the phagosomes. Macrophages are also scavengers that will scavenge through your body, finding bits of cellular debris and finding damaged cells. There's also a third type of endocytosis called receptor-mediated endocytosis. And cells can pick up specific types of molecules through receptor-mediated endocytosis. Sometimes you see this called clathrin-dependent endocytosis because clathrin is involved in forming those vesicles. In receptor-mediated endocytosis, we use receptors. Those receptors are on the outer surface of the cell, and when something binds to it, it triggers the cell to start to invaginate and form a vesicle. This is how mammalian cells can ingest hormones, growth factors, serum proteins, enzymes, all these things that can bind to receptors, cholesterol, antibodies, iron, viruses, and bacterial toxins. In receptor-mediated endocytosis, first you have a specific molecule or a ligand that binds to the receptors on the outer surface of the cell, and those receptor ligand complexes will diffuse laterally and encounter these specialized coated pits. The coated pits are made to collect and internalize these complexes. In your cells, in a typical mammalian cell, coated pits take up about 20% of the total surface area. So it's not most of the surface area, but it is a good big percentage of it. Then the accumulation of receptor ligand complexes in the pits will trigger the accumulation of additional proteins on the cytosolic surface of the membrane. And these are called adaptor proteins, clathrin and dynamin, and they help invaginate this pit here. And eventually the pit pinches off and forms a coated vesicle. You can see it's still got things like clathrin around the outside of it. The clathrin coat gets released and leaves an uncoated vesicle. The uncoated vesicle fuses with an early endosome. And within this, the transport vesicles can carry material to the late endosome and then to a lysosome for digestion. They can recycle receptors back to the plasma membrane, or they can transport material to the plasma membrane on the opposite side of the cell, what's called transcytosis. All right, so that was a fun little introduction to endocytosis and exocytosis. We still have probably one more video lecture to get through this chapter, and so I'll see you soon. Bye.